Hi, Richard Sever from Cold Spring Harbor Lab. I'm here at the virtual 2021 Symposium at Cold Spring Harbor Lab, a sort of strange event for everybody. This is normally a place where everybody's meeting at the bar and in person, so it's a bit of a change, but I'm delighted to say I have Gary Ruvkun here, who gave a fascinating talk last week, and we'll chat a little bit about the work you've been doing recently. So welcome, Gary, and thanks for being here.
Yeah, it was fun. It was a fun meeting for me because I'm not deeply in the biological timing meeting in the sense of what most of the people at the meeting work on. So it
Right.
was very different for me. And
Well, I mean, I thought your talk was fascinating because it seemed not so much about timing as editing, and it was almost like a move over CRISPR, nucleic acid editing is
yeah,
the old news, protein editing is the new thing.
yeah. Well, I do that tongue in cheek because
But
CRISPR has, is such a phenomenon. And Jennifer Doudna is an old buddy of mine, so I, I,
oh
I,
right,
li-
okay.
I like to,
So there's an interesting subtext there.
yeah.
Well,
It's-
I mean, I mean, well, let's sort of kind of plot a trajectory to that editing discovery you've made. I mean, I thought was what was kind of interesting was you did this talk that kind of ends up with kind of virology and protein editing, but starts with looking at proteasomes in worms and the rather provocative statement that you don't believe in hypothesis-driven research, which I kind of liked, and I'm sure there's a few people in genomics who would agree with you. But so what was the starting point then? You were looking at expression of proteasome subunits and what what regulated it?
yeah.
How did
So
you come to that?
I should amend my statement. I don't believe in correct hypothesis-driven science. I believe in incorrect hypothesis-driven science. So a wrong hypothesis is great. A right one is really boring. Uh, so, oh, this is terrible. Um, so, uh, over the last um, I don't know, maybe it's around starting in around 2005, I would say. So it was 16 years. We were doing experiments on lifespan in the worm. What are the gene inactivations by RNAi? This was right after RNAi was sort of discovered and became a genomic tool. What are the gene inactivations that will lengthen the lifespan of a worm? And uh, we kind of rediscovered what we had discovered earlier, that insulin signaling was important. But then we found a whole bunch of gene inactivations in what turned out to be essential genes, things that are really highly conserved genes that uh, most organisms have, like the ribosome or translation factors or the proteasome or the mitochondria. And if you inactivate those genes late in life, not if you do it early, they're dead. But if you do it late in life, it extended their lifespan, and it was kind of a it was bothersome because it's like, how do you why would you know inactivating a core thing like translation, why would that extend lifespan? And it was when Sean Curran, who was a postdoc in my lab at that point, he was doing these lifespan assays on worm. It's a 3-week experiment where you give treat them with the double-stranded RNA E. coli, so they're eating E. coli that uh, inoculates them with the double-stranded RNA that knocks out the gene, and that lowers their gene activity, and then you can assess the lifespan. And he would find that these animals live longer, but he had a technical problem that when he would do something like translation, elongation factor for translation RNAi, a lot of the animals uh, would sort of move away from their E. coli food and actually commit suicide by crawling up the side of the agar dish. And he kept losing animals to suicide essentially. And so and it was a technical problem, but as we talked about it, it was like a light bulb went off, and it was like, well, no, 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 these animals feel nauseous. they're eating bacteria that makes them sick because it's inactivating their ribosome, and they have a surveillance program that monitors their ribosome because over the last 3 billion years, or 2 billion that eukaryotes have existed, um, uh, uh, microbes have been attacking our translation apparatus. And so if you take a poison that poisons your ribosome, you feel crappy. And by the way, you're not hungry. And
Right.
and whatever you ate before you ate that, you think, I'm not going to eat that again. Not shrimp, never again.
Right. Right.
I've eaten
Teenager
octopus
with vodka.
once. I ate octopus once in Greece, felt horrible, and have not had octopus since.
Right. Yeah.
Anyway, so that got us going on a whole program of, well, how does it know that the ribosome's not working right? What are the signal transduction? Is that involved in human anorexia? Is this, can we make anorectic worms? So the lab took a pivot away from aging towards essentially innate immunity. And so the proteasome project started with Nick Lauerbach coming as a postdoc, and I was sort of pitching it to him as, look, we've already done this with the ribosome and figured out some of the signaling apparatus. The proteasome is similar in being ancient and a real part of a defense pathway, because proteolysis is a very good way to defend yourself against virulence factors and things like that. So I was envisioning his screen for proteasome homeostasis was going to be similar to the ribosomal screen that we had done, or the mitochondrial screens. And his genetics proved that I was completely wrong, that the proteasome is surveilled in an entirely independent axis that is all about ER stress and probably related to viral stress, but not so much related to bacterial defense.
Right. So this is the point at which up pops the skin-1 transcription factor, is that right?
That comes out, and the biggest surprise was the PNG1/NGLY1 deglycosylation enzyme that came out of the same screen, as working in the same pathway as the skin-1 transcription factor.
So yeah, so in your talk, you said that there is the NGLY1 is this glycosylase that regulates skin-1. So skin-1 is the first thing, that the transcription factor that's controlling new proteasome component synthesis. Is that how it works?
Yeah. So it directly binds to the proteasome genes and ups their expression. And in fact, this was discovered before us, right? So the skin-1 was a candidate gene that came out of looking at what does it take to increase proteasome gene expression when you treat a mammalian cell with bortezomib. We didn't discover this at all. And so the human homologue of that came out of that genetic analysis. So skin-1 sort of folded into our analysis, and what came out of our analysis was the NGLY1 and the protease called DDI1 that clips skin-1.
Right. So skin-1's basically sitting there in the ER doing ER associated decay. Then along comes this glycosylase. And I mean, for me, that was when the story got really interesting, because it was like, it's doing some deglycosylation, but that's not important right now. What's important is what happens as a consequence of that. So can you explain that, thinking about this glycosylase, but actually it's like there's something much more interesting going on, well, at least to non-glycobiologists.
Yes. So that what's interesting is that this deglycosylase had been purified by Tadashi Suzuki 20 years earlier, and was being looked at as a sort of how cells deal with aggregated glycosylated proteins and sort of aberrant glycosylated proteins as a kind of a degradation pathway. So it was really being looked at as, well, if you're going to send a protein to the proteasome, you need to deglycosylate it so that it can kind of get ground up. It would clog up your garbage disposal. But it had been noted years ago that that it was in the process of deglycosylating N-gly1, as it's called in mammals, deamidates. So it actually uses water to cleave the amide bond, and the amine goes with the sugars, wherever it is the sugars go. And then the protein has been edited with an asparagine to an aspartic acid edit. But people were thinking about it in terms of protein degradation, and that the only time you would ever deglycosylate something was in the process of degradation. But what our genetics said was that the N-gly1, or PNG1 as it's called in the worm, was necessary to activate this transcription factor, that the error was not that it was inactivating it, but that process was activating it. And so that's where Nick Lehrbach decided, well, then if N to D is what the enzyme is doing, let's change in the genome N to D and see if we can bypass the need for PNG1 or N-gly1. And when he engineered those N to D substitutions into the target gene SKIN1, you now bypassed any need for N-gly1. And what was interesting is that if you then compare in genome sequences, you can see those N to D changes in comparing C. elegans to other Caenorhabditis and even other parasitic nematodes, which are much further away. You can see these N to D changes. But one of the points I made in my seminar is overinterpretation of data is a good thing. Not that Nick overinterprets data; he's just the opposite. But his supervisor, me, is really into overinterpreting. So, we saw one example of an entity change in evolution, and that was it. That was enough to prove the case for me. But it wouldn't peer review. It wouldn't stand up to peer review.
So, I mean, mechanistically, what's that doing? So we're basically saying the sort of the glycosylase aspect is irrelevant. You've got to change from an asparagine to an aspartate. And one thing that I made, which I think you talked on as well, was I was like, asparagine, aspartate. Oh, that's changing the sign. Acid blobs. Don't we have those?
Acid blobs. You see, but you have gray hair,
right?
So this was all. You have to understand that I started my lab in 1985, and a lot of my best buddies. Roger Brent started his lab next to mine. So you had two highly immature assistant professors chatting with each other. We'd known each other as grad students too, right? And Brent was Mr. Acid Blob, transcription activation domains, fuse something to a DNA binding domain and get it to work. So I was indoctrinated into acid blobs. It's probably been lost in history. Does anybody else think about acidic regions of transcription factors anymore?
I don't know. I think when I was an undergraduate, that was what we were told, was that you basically took a Gal4 DNA binding domain and then you stuffed loads of acid stuff next to it and a transcription factor.
Yeah, so I must be older than you. I'm 69 years old. You're probably 63.
But yeah, so I mean, is that so? You then look on evolutionary terms, and you start seeing this elsewhere. I mean, you say
Yes.
You're extrapolating from one, but you looked at other things, and up pops this.
Well, in the human variation, you could see many more examples of these entity changes in glycosylation sites, and then we sort of genomicized it, and we said, okay, looking in the worm genome. At proteins that spend some time in the ER because they have ER annotation. what's the frequency of N to D changes if you're in an N-glycosylation site, which is N, X, S, or T. And it was 3 times what it is for any random N. So it said that N to D changes are common over evolutionary time and common in N-glycosylation sequences. And that's where we segue to the viruses. And just to give a sense of the historical moment. So I start looking at this in like March of last year because we're stuck at home and all I can do is computational work. And I'm, scared to death of COVID. And so, but the best way for me to deal with that psychologically is to say, well, I know that I'm going to look at the glycosylation of this virus differently than anyone else. And it's a skewed look that could either be misguided or brilliant. And it's still somewhere in between those two.
I was about to say, have you decided which one of those things it is?
It's both.
But you find this sequence in SARS-CoV-2 then.
Yeah, so then the glycosylation sequences are well known in SARS and CoV-2, and so then now you do an alignment of all these viruses and say, okay, where I can align it, do you see evidence of N to D changes happening in viral evolution? Mm-hmm. And so we see lots of good examples of this where the only thing that's there is either an N or a D. Right? It's not like it's mutating to anything. It's not like the virus doesn't care. It's like for some hosts, and if you look at the annotation, bat comes out more often when there's a D. And so the rationale, again, it's a house of cards, but the way I think about it is that, maybe the bats lead a more desiccated environment, right? They have a super high metabolic rate, crazy high metabolic rate. They're living in their stinking caves. And, maybe for them, it's better to have the genome give you the N to D rather than the glycosylation, deglycosylation cycle.
So is the idea then that there's a kind of constraint on the protein that means you've got to have an asparagine or aspartate, nothing else will do. It's got to be one of those two things.
Yes, and there might be a reason why the aspartate is useful to the virus. Mm-hmm. So and this is where the human genetics comes in. So, when Nick discovered in my lab that NGLY1 is important for this pathway, that's the deglycosylation enzyme. There was a human literature, very small literature, like 5 papers on human NGLY1 disease. And actually there's a whole New Yorker article about the parents, because they're very proactive parents that started a foundation to try and figure out how this disease works. And so Nick and I became part of this group of maybe 30 different labs across the world that think about it. And when they did a clinical characterization of maybe a third of the kids who have this, so these kids are really sick. They're wheelchair bound. They have epileptic seizures. They produce almost no tears, so they have corneal scratching and such. It's sort of the real hallmark is alacrim, a lack of production of tears. There's some evidence that they have problems of aldosterone production in the adrenal gland. So another gland is kind of aberrant. And the parents report these kids don't get colds and flu.
Right.
And it's a little anecdotal, right? Because it's like parents saying, no, my kid never gets sick. And, does that mean anything? But when they did the clinical characterization of these kids, they noticed all of them are vaccinated, like all kids are, and their level of antibodies to rubella was way high. So they're somehow mounting a better than normal immune response to a vaccine, which kind of fits with them being resistant to a virus.
And so they're stuck with the asparagine. these kids.
That made me think it's maybe tied to the asparagine, that the viral life cycle includes glycosylation big time, right? Because glycosylated, extracellular secreted proteins, the spike proteins, the related proteins of the hemagglutinin in influenza virus, these are all part of how they interact with their receptors or try to hide from immune system. It's part of the battlefield between viruses and hosts. And so that just made me think that this asparagine might be salient to a viral infection, and we should be thinking in those terms. Then we started to intersect, look up in various databases. Remember, everything we're talking about, this N to D editing, is in a really tiny corner of biology. It's the worm. It's in a corner of worm biology. It's sort of how the proteasome is regulated. But you can go to the cancer dependency map, which is a case, the Broad does this, where they have 1,000 different cancer cell lines, all different kinds of aneuploids and, glial cells and bone marrow and, you name it. And they take 1,000 different cell lines and they do NGLY1 CRISPR, NGLY1 RNAi, and they find 100 cell lines that really grow very badly when you inactivate NGLY1. Mm-hmm. And then they do this with every other gene in the genome and ask, well, who looks the most like NGLY1? Right? They do this for all genes. And if you look at the correlation coefficient, the cancer dependency map rediscovered what Nick Lauerbach's great genetics discovered, that NGLY1 works in a pathway with SKIN1 and the DDI1 protease. The cancer dependency was absolutely coincident. It was really glorious. And we don't understand why that is, but it probably says something about aneuploidy. The hallmark of these cancer cell lines is that they have weird gene dosages. They amplify this and they duplicate that chromosome region, and protein dosages are really impacted in multi-subunit complexes like the ribosome and things like that by aneuploidy. So these guys are really, really sensitive to proteasomal homeostasis. Same thing happens in viral infection. The proteasome is really challenged when a virus is putting together all these virions and not all of them get assembled well, and the virus is not really in the business of trying to keep the cell happy. It just wants a big burst, and so it made sense that this same pathway would be part of an antiviral response. And other people had done hepatitis screens, and NGLY1 came out of an RNAi screen for hepatitis resistance. So it added up to a case could be made that it was important.
Right. Well, I mean, it's a fascinating story. I mean, it seems to me that, it's a great advertisement for not having a hypothesis in the first place, because you go from, something that you thought might apply to ribosomes that didn't apply to proteasomes, ends up telling you something about cancer and virology and potentially all manner of other things. Where are you going to go next? I mean, you kind of presume that the road is forked in many different places, and there's a whole
yes, yeah,
yeah.
Well, to me, this was the first project in my lab where we did sort of big time genetic screens where there was no mapping. So you have to understand the history of genetics in worms and flies, not so much in yeast. Has been, you do a screen, you get a mutant that you like, and you spend the next year in hell doing linkage mapping, with your Excel spreadsheets, and they must have done it with graph paper in the past. I don't know what, before Excel, there must have been people must have had tables in notebooks and things.
Yeah, I think
it must have even sucked worse.
They didn't have Excel.
Can you imagine? And I have to say, I was a very mediocre geneticist in that era, because it's a bookkeeping it demands bookkeeping skills. Now, in this case, Nick did a screen for animals that turn green in a proteasome inhibition, or failed to turn green, got green or ungreen animals, picked 100 animals, did the genome sequence, and boom, 3 weeks later, 6 alleles of DDI1, 2 alleles of PNG1. You instantly figure it out. And did we have a hypothesis that those genes would be involved? No way. Did we learn that they were involved? Yes. And we were taught by our genetics.
Well, I mean, you said in your talk that you like the new genetics a whole lot more than you like the old genetics. So I'm beginning to understand why. Gary, it's been fascinating talking to you. So thanks very much, and good luck with all the future work on this.
Thanks.